Microbial genetics, DNA to RNA to protein. As we go through the three different mechanisms that utilize DNA, RNA, and the eventual production of proteins, we're going to find that there are three stages or phases for each mechanism. The mechanisms are replication, transcription, and translation. Each one has an initiation stage, an elongation or polymerization stage, and then a stop or termination stage. That's going to occur for each replication, transcription, and translation event. The first mechanism is replication, or what do cells do with DNA? Polymerization of DNA is an important process that a cell must do to generate the next generation or a copy of its hereditary material to pass along in cell replication. We describe replication as DNA-dependent DNA synthesis. In other words, we're going to have an original DNA template from which we can copy another piece of DNA. That's replication. Now, DNA is going to serve as the master blueprint. It really is not sort of the working blueprint. If we need a working blueprint that we are actually going to be able to use and interpret, just like a typical blueprint when we're creating a building, then we're going to need a working copy of a nucleic acid, but it's going to be in the form of RNA. So DNA is the master blueprint, but the working blueprint that we're actually going to interpret is in the form of RNA. And to make RNA copies of DNA, we're going to use a mechanism called transcription. So we define transcription as DNA-dependent RNA synthesis. We're going to need a DNA copy or DNA template from which we're going to make RNA copies. So please make sure that you can distinguish replication from transcription. What is DNA? DNA is formed from essentially three different components. We're going to use a nitrogen base, purines and pyrimidines. The purines will be adenine and guanine. The pyrimidines, thymine and cytosine. We're going to find that eventually we're going to pair these purines and pyrimidines together so that we have one purine hydrogen bonding to one pyrimidine. Which one bonds with which? Well, it's going to depend on quite a few things. But ideally, when we have the proper kind of hydrogen bonding between the purines and pyrimidines, we're going to find that guanine will always hydrogen bond with cytosine in the essential ideal formation of DNA, and thymine will always hydrogen bond with adenine. We're going to see that ideally we have two hydrogen bonds for the AT base pairs and three hydrogen bonds between the GC base pairs. That's how we describe them. When we pair one purine and one pyrimidine together, we call them nitrogen pairs or base pairs or nitrogenous bases. There's all kinds of phrases or terms that we use to describe a pair of nitrogen bases, one purine and pyrimidine, typically found in nucleic acids like DNA, and eventually we'll see the same or similar kinds of base pairing in RNA with one slight substitution. Notice the reason we call them nitrogen bases is because they're loaded with nitrogen atoms. It's not all carbon on those molecules. There are quite a few atoms of nitrogen, and that's where we get the name or the phrase nitrogen bases. Now, in addition to nitrogen bases, we're also going to use a sugar when we're forming nucleic acids. If we're creating DNA, we're going to use deoxyribose. So, one molecule of deoxyribose and one nitrogen base. We're starting to essentially assemble some of the monomers or building blocks for nucleic acids of DNA. If we're creating RNA, we're going to use ribose plus a nitrogen base if we're starting to create the monomers or building blocks for RNA. Again, notice the subtle difference between ribose and deoxyribose. They're not the same sugars. There's only one atom difference between the two, but that makes a huge difference in how the sugar will be used. So, ribose has a hydroxyl group as one of its functional groups. For deoxyribose, we've substituted simply a hydrogen for that hydroxyl group. Note in the little figures how the different pieces or parts of these two sugars are highlighted. Tiny little difference makes a huge difference in how the sugar will be used. The last component of our building blocks for nucleic acids would be a phosphate group. And actually, it's going to be 3 phosphate groups. Once we've put all of these parts together, we have now assembled a nitrogen base, either adenine or thymine or guanine or cytosine. The sugar, if we're creating DNA, it's going to be deoxyribose. If it's RNA, we're going to use ribose. And then if we're using free monomers and we're essentially creating the building blocks for nucleic acids, then we'd use 3 phosphate groups. When we assemble these components, the phosphate groups, the sugar, and the nitrogen base, we now have the monomers or building blocks for nucleic acids. We call those building blocks nucleotides. So we're going to use nucleotides to make nucleic acids. Now, the history how scientists determined how these different pieces work together, and in fact that DNA was the molecule of heredity, is quite a fascinating history. Some of it starts with a scientist by the name of Erwin Chargaff. And ages ago, many decades ago, scientists knew that if you took the nuclei out of a cell and you purified the chemical, you'd find this kind of sticky stuff inside the nuclei of various kinds of cells. And eventually scientists identified this stuff as DNA. Now, they eventually also figured out the nitrogen bases that were found in DNA, the AT and the GC. But they really couldn't determine, was this really the molecule of heredity, or how was it used, and how was it arranged? It was quite perplexing. What Erwin Chargaff helped to figure out is just how much of each kind of nitrogen base is in each kind of cell. He did this by taking samples of DNA from all kinds of species of organisms and simply measured how much adenine and how much thymine, how much guanine and cytosine were in all of these different kinds of cells. Lo and behold, what he found was that there were certain ratios or percentages of adenine and thymine, guanine and cytosine. And it was consistent every time we took a sample from that representative organism. So if you notice on the chart, for the percent distribution, he found that whenever he measured how much adenine was in his DNA, he had roughly the same amount of thymine. When he measured how much guanine was in his DNA, he had roughly the same amount of cytosine. Now, it didn't tell him anything about how they were bonded together or how they were interacting. All he could determine were relative amounts. But it really is one of our first pieces of the puzzle to figure out how DNA is constructed and how the cell is going to use it. We're going to see that to create polymers of DNA, we're going to link the different nucleotides together alternating sugars and phosphate groups. And it's the phosphate group that's going to be used to link the sugars together. Notice the arrows are pointing to the oxygens that are part of the link that holds the sugars together. Each phosphate group creates one ester bond to two neighboring sugars. Since we have two phosphate groups, we say that we have phosphodiester, di meaning two ester bonds. So that phosphate group helps to link one sugar after another, until we have a whole chain of nucleotides held together by phosphodiester bonds. The other thing you'll notice on this slide, at the top of the picture, there's a 5 with an apostrophe. Well, the scientist's word for the apostrophe is prime. So we say we have a 5' terminus at the top and a 3' terminus at the bottom. It's an orientation, just like north and south. So we describe the orientation or the directionality of this polynucleotide chain as 3' at the bottom and 5' at the top. A 3', 5' phosphodiester bond all the way up and down the chain of nucleotides. One of the scientists that was quite instrumental in determining the three-dimensional structure of DNA was Rosalind Franklin. In the late 1940s, she was given the task of taking one particular form of DNA and examining the structure to determine its three-dimensional organization. Using a technique called X-ray crystallography. X-ray crystallography allows a scientist to view the individual atoms within a structure. And Rosalind Franklin was particularly good and quite expert at purifying and creating exquisite crystals of DNA, which were then exposed to X-rays and producing what are called X-ray diffraction patterns. Now, it's quite amazing, but using mathematics, and realize that she didn't have fancy computers and did a lot of the calculations by hand, but taking careful measurements of the different dark and light spaces from her photos of the X-ray diffraction pattern of DNA, she was actually able to determine that DNA was a double helix, that the sugars and the phosphates ran along the outside of the helix like a ladder, and quite a bit about the different structures and structural components of DNA using her X-ray analysis. Now, as it turns out, her information about the three-dimensional double helix structure of DNA was quite integral and quite important for scientists to eventually figure out the overall structure of DNA and how it works. But unfortunately, when scientists were being given Nobel Prizes for their contributions in the scientific revelation of how DNA works and the structure of DNA, she unfortunately had already passed away, dying of a cancer that was probably the result of her overexposure to the ionizing radiation of X-rays that she worked with during her studies and using X-ray crystallography techniques. A few other scientists who were also vital and critical to the determination of the structure of DNA and essentially how DNA was assembled were James Watson, Francis Crick, and Maurice Wilkins. Watson and Crick really did manage to synthesize the whole picture. They used information from various scientists. They had a chance to gather up the information from Erwin Chargaff about adenine and thymine ratios, guanine and cytosine ratios. They got essentially a sneak peek at Rosalind Franklin's X-ray diffraction patterns. and her interpretation that DNA was a double helix. They had information from biochemistry textbooks and some of their biochemistry colleagues about how thymine and adenine could hydrogen bond and how guanine and cytosine could hydrogen bond. Then putting all those pieces together, they eventually created models, three-dimensional models using paper clips, popsicle sticks, you name it, pieces of paper. They just created all kinds of possible models to explain how this particular structure of DNA would look. And sure enough, eventually, they did get it all right. And that's, of course, what Watson and Crick are remembered for today, that they eventually determined and reported the actual structure of DNA. Well, that was a good start. Now that we have the structure, how in the world is it going to make a copy of itself? Or in other words, how does DNA replicate? So how does DNA replicate? Well, we'll see that that was probably one of the first answers that was needed. What kind of method does a cell use to copy DNA from one cell to the next? A really important step. Three possible models of DNA replication were proposed. Of the three, two of the three always seemed possible to me. The third one just didn't make sense at all. The first proposed model of DNA replication was conservative. We take an original piece of DNA and then make a completely new copy somehow from the original piece. And so that's what's illustrated in the first column, where we have the original DNA in dark blue and the newly synthesized DNA in light blue. The second possible model that was proposed was semi-conservative. In the semi-conservative model, we start out with an original double-stranded DNA template, just like we did with the conservative model. And we're going to take the two halves of this double-stranded DNA apart, make one half new and one half old, and then just continue to do this process of separating the two strands of double-stranded DNA, synthesizing a new copy alongside the old. So we always have half old and half new. That's semi-conservative. And then the third possible model that was proposed was dispersive. In the dispersive model, each strand of both daughter molecules contains a mixture of half old and half new. Now, this model really never did make any sense to me. I don't see how. It was difficult to propose a mechanism or a way that these bits and pieces would somehow get bonded together and still have fairly true fidelity or copying. It seems to me the dispersive model would have been quite susceptible to errors or mutations, whereas the conservative and the semi-conservative model, maybe not so much. So let's see what did happen to figure out which of these models was the correct one. This slide illustrates what's often been described as the most beautiful experiment in science. It starts out with cultures of E. coli, and the cultures of E. coli were grown with food that contained a heavy isotope of nitrogen called N15. Now, N15 is just a little bit heavier than N14, which is the more common form of nitrogen. It simply has one more neutron in its nucleus. But it's heavy enough that we can actually find it and separate it from N14. So we can measure how much and where the N15 is. So we feed the E. coli some N15 containing food, for a little while. We stop the growth, extract the DNA, and then visualize where that DNA is. We can essentially separate the DNA in an agarose gel, and then we can color it or stain it. We can take photographs of it. That's what the band pattern is. And we can also scan that band with another kind of photographic material or instrument and sort of graphically depict the width and the density of that band of DNA in the agarose gel. Then we keep the E. coli growing, only now we switch the food. And instead of nitrogen 15, we're going to start feeding the E. coli nitrogen 14. Now, if it's semi-conservative replication, we should start to see essentially different bands or separate, distinct sort of groups of DNA, band patterns, in our agarose gel. If it's conservative, we're going to see essentially one tiny little bit of N15 DNA and then bigger and bigger bits of N14 DNA, but there's always going to be some tiny little bit of the N15 all along the way. If it's semi-conservative, we're going to find that it's that N15 piece of DNA is harder and harder to find because we only have one copy and it sort of gets blended in since it's half N14, half N15, half old and half new. And sure enough, that's what Matthew Maselson and Franklin Stahl, who performed this experiment, found out. That every time the DNA was allowed to undergo another growth cycle, and then the DNA was extracted and separated in agarose and then photographed, we found that there was more and more of the N14, and then we still had that hybrid N15, N14 DNA, half old and half new, that was still present, just as we'd seen in that second replication cycle. Using this particular procedure, Maselson and Stahl were able to absolutely confirm that DNA replication is semi-conservative. We're going to take the two halves of the double-stranded DNA ladder apart, and then we're going to line up new complementary nucleotides along the original half of the ladder that's sticking out, and synthesize sort of a hybrid, synthesize half old and half new every time we go through a cycle of DNA replication. Along with the double-stranded DNA structure of DNA, notice that, again, we have that 3', 5' orientation. The 3', 5' are actually referring to the carbons on the sugars, deoxyribose, and where the carbon is located, to which we can either attach a hydroxyl group or a phosphate group. And what we find is that the number 3 carbon of the deoxyribose is the carbon to which we're going to attach a hydroxyl group, and the number 5 carbon is the carbon to which we're going to attach a phosphate group. So scientists refer to the 3' hydroxyl, 5' phosphate orientation of DNA on both sides of the DNA ladder. Notice that they're going in opposite directions, which is described as anti-parallel. If you look at the picture, you can see that on one half of the ladder of DNA that's been flattened out in the illustration, the left side, which is highlighted in pink, has a 5' carbon with a phosphate group at the top and a 3' carbon with a hydroxyl group at the bottom. On the opposite side, backlighted in blue, it's just the opposite. The top, the carbon is the 3' hydroxyl. The bottom, the carbon is the 5' phosphate. What often is left out, and students get confused, is that the geometric shape of the deoxyribose sugar, the little pentose illustration that's used in this drawing, actually means that there's a carbon at every angle, at every corner or angle on that sugar in that little 5-sided pentagon drawing is a carbon at every little angle, and then there are hydrogens and hydroxyl groups at various places. So the little 5-sided cartoon drawing of the deoxyribose sugar really is just a very minimalistic, very rudimentary structure of deoxyribose. If you want to get a better idea of what atoms are located in all the different places on the deoxyribose, so you get a better idea of where the functional groups are attached, please refer back to one of the original slides at the beginning of this presentation. Now, there are some very important enzymes that are going to be responsible for DNA replication. In fact, we're going to have a whole grocery list of enzymes that we're going to need to perform DNA replication. It's a highly complex process, and it has to be very well controlled so we don't have any mistakes. Well, maybe not too many mistakes, because we have to have a few from time to time. But we'll talk about mutations in another presentation. But the key enzymes responsible for a lot of the work that's undertaken during DNA replication are DNA polymerase 3 and DNA polymerase 1. Now, DNA polymerase 1 really was the first polymerase to be discovered, and in fact, E. coli turned out to be an excellent example or role model to study DNA and DNA replication decades ago. It's very easy to handle, easily grown, and it turns out that most of the DNA that's extracted from E. coli is almost all pure DNA. Not a whole lot of extra stuff in terms of other kinds of molecules attached to the DNA. So it really was a good starting material and a starting organism to use when scientists tried to figure out what are the hereditary molecules of organisms, how they replicate, how do cells manipulate and use the nucleic acids. And so, as it turns out, DNA polymerase 1 is a repair polymerase. It's going to mend things and make things better. Well, there's a lot of that kind of enzyme in a cell because there's a lot of repair that occurs. But when scientists really tried to make large molecules of DNA, they found that there actually was an enzyme that had the huge task of DNA replication. And the replication enzyme is DNA polymerase 3. Now, a polymerase is simply an enzyme that makes a polymer. What kind of polymer? Well, it's either DNA polymerase or RNA polymerase. And when we tell what kind of polymerase it is, that tells us what kind of molecule we're making. So a DNA polymerase is going to be used to make DNA. DNA replication is going to need quite a few enzymes. We're going to establish a replication fork. In other words, we're going to take the two halves of the DNA ladder apart. Notice that one half of the original DNA ladder starts with a 3' terminus, and the other half of the DNA ladder starts with a 5' terminus. The original DNA is in gray, and any new material is either in light yellow or sort of a reddish orange. Now, if we take a look at the left side, notice that it says topoisomerase nix DNA to relieve tension from unwinding. Now, a topoisomerase is an enzyme that's responsible for either introducing supercoiling or relaxing supercoiling. And what is supercoiling? Well, supercoiling allows a cell, or nature, to take a really long, stringy molecule like DNA and zip it up or compact it or compress it into a sort of tangled mass that's really, really tiny and doesn't take up much space at all. In fact, DNA must be supercoiled in order to properly function. And in order for us to replicate it, we have to relax the supercoiling in order for us to get started with DNA replication. So that's where we get started, with a topoisomerase that's going to relax the DNA and allow the next enzyme, which is labeled number 1, called helicase, to separate the two sides of the DNA ladder and create what we call a replication fork. Now, it's a two-tined fork, but it's a fork nonetheless. Now, believe it or not, there's more going on in this process than what's even on this slide. What's not shown on this illustration is that when helicase opens up the double helix, there are actually single-stranded DNA-binding proteins that keep and hold this fork apart just long enough for some additional enzymes to come in and do their job. One of the enzymes that's responsible for the majority of DNA replication is DNA polymerase III. Notice at the top strand, DNA polymerase III is going to synthesize or pair up new complementary nucleotides with the original DNA and create one nice long entire piece of complementary new DNA with the old DNA. So we have half gray and half sort of reddish. We describe this as continuous synthesis of the leading strand. So the leading strand is that bright orangish red new DNA that we've now created this nice long piece of double-stranded DNA, half old and half new. But if you look at the bottom half of the picture, we can see distinctive differences. And DNA replication on the bottom strand is only occurring in bits and pieces. In fact, the scientist who was the lead investigator of the group who determined that DNA replication does not occur exactly the same using the old template that starts with the 3' end versus the old template that starts with the 5' end was Dr. Okazaki. When he allowed E. coli to grow and they stopped synthesis or stopped growth every so often and extracted the DNA, they found these huge pieces of freshly, half, semi-conservatively replicated DNA. And then they'd also find some little bits and pieces or fragments that clearly told him or showed him and his colleagues that DNA replication on the 5' template DNA is essentially being created in small fragments or discontinuously. We describe this as discontinuous synthesis of the lagging strand. We're going to need an enzyme called primase to create an RNA primer on the lagging strand. And that's going to allow DNA polymerase III to bring in complementary deoxyribonucleotides and fill in as much as it can until it meets another RNA primer. Once DNA polymerase III has done as much as it possibly can do, then we're going to need the help of DNA polymerase I. DNA polymerase I is going to remove the RNA primers, and it's going to bring in complementary deoxyribonucleotides to fill in the gaps where the RNA was. Now, once we've filled in the gaps, and we now have, hopefully, a nice, almost complete DNA piece on the lagging strand, now we're going to need to seal up that sugar phosphate backbone. And to do that, we're going to need DNA ligase. DNA ligase as the name implies, is going to ligate or sort of sew together all of those nucleotides that were now lined up and assembled on the lagging strand. And it's even used on the leading strand too. And we now have hooked together or linked together all the deoxyribose sugars with all the phosphates, and all those nice phosphodiester bonds are all now nice and intact. And we now have complete, hopefully, identical copies, two, that match in terms of their nucleotide sequence the original DNA that we started with. So here we have a close-up of this elongation phase, of the fact that DNA polymerase III is pairing up the ideal, proper, complementary nucleotides from the new strand, bringing it and lining it up with the original template strands. So that if we have an adenine on the original template of DNA, DNA polymerase III will help line up a nucleotide that has thymine on it. If we have cytosine on the original template strand of DNA, DNA polymerase III is going to help line up a nucleotide with guanine on it, so that we can have the proper hydrogen bonds forming between the old and the new complementary nitrogen bases. Now, also notice that the free nucleotides, the nucleotides that are being used in this synthesis or the elongation of the new half of the DNA, are in the triphosphate form. Those two phosphate bonds, a phosphate bond, when we link the two phosphate groups together, it's almost like putting a spring, taking a nice tight spring and compacting it. All of those negative charges on all those phosphate groups, they really don't want to be next to one another. We sort of forced them together. So when we remove those phosphate groups, it's almost like taking a spring that we've compressed by squeezing the two ends closer together. And then if we let go of that spring, the spring just goes pop, just sort of stretches really quickly. There's that mechanical motion or mechanical movement of the spring when we release it after it's been compressed. Well, imagine the same thing happening with the phosphate groups, except instead of mechanical movement, we have the potential for free energy to be used to do work. And that's what happens. We're going to use the potential chemical free energy released from the phosphate groups on the free nucleotides to help link them together when we're polymerizing or synthesizing DNA or replicating DNA. So this is DNA replication, when we're making a polymer half old and half new during semi-conservative DNA replication.